Welcome everyone. We will give a couple of minutes for everyone to join.
It's my great pleasure to welcome you all to this King's College London Engineering Association lecture, which is actually the 7th in a series, the annual lecture that was instigated in 2015 when the engineering department at King's was closed. But the engineering alumni could not accept that it would be sidelined in higher education in a modern society. And so, of course, now I'm delighted that the new engineering department is fully operational, despite all the challenges of the pandemic. So we are now into a different era with the history of our lecture. And this is the first time that we've used the web-based platform. So it's very pleased to tell you that we have actually 116 registrants for this webinar. And there's 100 alumni in a dozen different countries. And that, I'm told, includes somebody dialing in from Japan. Now, just before we get into the lecture itself, let me just touch quickly on the formatting. Many of you are probably very familiar with the Zoom format. And thank you to those who may have already submitted questions in advance. But during the lecture, you can post a question using the Q&A button that's at the bottom of your screen, which will then be collated by the panel. And also, if you're seeing that question come up and you like it, you can press the little thumbs up button, which would indicate that that question is most popular amongst all the questions that may arise. Now, we all know in this country how yesterday was the National Day of Reflection for a year with this terrible pandemic that we've had. So could there be a more timely and topical subject for this year's lecture, which is the discovery and scale up of vaccines and novel therapeutics in the age of artificial intelligence and robotic automation? The timing is just incredible. It gives me therefore great pleasure to introduce King's Professor of Sustainable Manufacturing Systems, which is Htrz Makatsoris, to give this year's lecture. And we're also very pleased that Professor Makatsoris was recognized by the Royal Academy of Engineering last year when he was awarded the President's Special Award for Pandemic Service. So I think enough said by myself, and I'd now like to hand over without further delay to Professor Makatsoris. Thank you.
Share my screen. I hope you can see it. Or not yet. I hope you can see it now, I guess. Right. Okay. Well, David, thanks very much for the introduction, and a big thank you to the alumni for inviting me to give this talk. It is really a great pleasure to present to you the work that we have been doing in my group, not just recently, but give you a kind of cross section in the topic of AI and robotic automation in the discovery and manufacturing, in particular, of materials which are very, very important to us, including vaccines, of course, more recently, and we now see why vaccines are really the cornerstone, really, of our civilization, as well as other materials, other therapeutic materials. So I'm going to give you a cross-section of our work here. It is, of course, a multidisciplinary collaboration with others. I have always been collaborating with other colleagues, experts in the field, complementing each other in research that basically involves these two things. So all my work has been, for the last 16 years or so, has been around these two strands. At the top is all about and if I can, I don't know if I can bring a pointer up just in case. So at the top here is all about the modeling aspects, the understanding aspects of how we can design and make these materials. But crucially is this bottom bit, which is all about figuring out ways of making them much more efficiently, much more repeatably, and importantly at consistent quality and at rates which are high, but also, you know, exploring whether we can turn the problem of manufacturing on its head and make it, you know, cheaper and much more accessible as well. And there's going to be a bit more of that later. Now, in the context of vaccines, if we put these two things together, they do translate into responsive and agile manufacture of these materials. So my work originated with an observation. Okay, so if anybody goes into a synthetic biology lab and a chemistry lab for that matter, they will see a picture that looks like this. I don't know whose lab this is, but it does look a lot like all the labs I have seen, and to a large extent, to some extent, my lab as well. So you see, there is a lot of expertise there, but the key observation here is that there is a lot of manual work involved. So the question was, is this really the state of the art? Is this the best we can do today? So I thought, okay, maybe this is the killer application for robotics. And anyone with a robotics background or practice, they might have seen that in the last, in the 90s, and notice actually, robotics was, I mean, it did have a lot of application in certain industrial sectors, but it was really a technology that was kind of hard to sell. And especially in academia, it was really difficult to actually make the case why public money should be spent in that space. Of course, research had been progressed, and you know, now the research is focusing more on application areas that can really benefit from this kind of automation. And it was coming across the situation, the picture like the one we have seen. I thought, okay, maybe that's a good area or a good important problem to solve, because the vision is, can we actually make discovery, scientific discovery in the space in particular of chemistry and synthetic biology to look more like an Amazon Web Services type setup. Of course, it wouldn't be just computers, it would be other things, and we're going to explore what these other things might be. Because the thing is, every researcher, be it in a commercial laboratory or in an academic research laboratory, we are all aiming at impact, rapid impact. That means, how can our inventions find their way in practice, in practical application? And in the case, of course, for vaccines, I mean, the case is blatantly obvious, but there is a lot of other materials, a lot of other therapeutics, drugs that do have to find their pathway into the hands of the consumers, the end recipients, be it for important life-threatening conditions or to make us feel better and keep ourselves, in a tip-top condition, as it were. There is also an additional problem. Okay, the work that you can do in the lab is absolutely fine. I mean, one can come up with all sorts of ideas, be it manual. So that translates, of course, to a lot of effort, a lot of time going into it. But there is a very, very big barrier. How can somebody go from the laboratory having to deal with, in the case of vaccines, we have been working on for the last 4 years or so, and for quantities that are in the scale of just a few milliliter, and that's not the dosage, right? Is the quantities we're talking about to actually do the work and figure out, what is the agent you have made sufficient in a sufficient quantity to be able to do some measurements, to actually deploying it in the really, really large scale that is required to find its pathway to the market. And we have all heard in the news recently that the deployment of vaccines manufacturing is a very big constraint. And we heard stories about some of the deployment plans going smoothly, and then all of a sudden we heard about batch crashes, delays in deliveries, and all sorts of ongoing discussions between various recipients of these materials, where everybody now sees that there is going to be a shortage in supply, because manufacturing of these things is really, really hard. This is a really very high specification material, and the methods we're using right now, they do present still a challenge. These methods have not changed in a long, long time. I mean, somebody can quote, that they're about 100 years old or more even. So there's got to be a better way of doing it, for sure. Now, if we consider then manufacturing, and if we consider the discovery aspects as well, and all the steps in between, going from the discovery of the molecule that you are interested in to figuring out how to make it at scales, one can start thinking, somebody with an engineering hat on could start thinking, okay, this is a lot of options. How can I explore this very, very big and very complicated space? I will certainly need new tools, okay? And hopefully automation can help answer a lot of these problems, not all, but a lot of these problems. I mean, we have seen in drug discovery the use of high throughput robots, but again, the issue with this type of robotics, which are, from an engineering perspective, by the way, very exciting machines, the issue is that they are not still concerned with the step of manufacturing and scale up. It is all about making sufficient quantities in the microgram scale, milligram if you are lucky, just sufficient quantity to be able to measure what it is you have done. And then taking this discovery through to the entire pipeline all the way to manufacturing is still a very, very big problem. So can we combine it all with some sort of automation? So the question then is, all right, all that is absolutely fine. What do we have to do? How can we develop such an automated system? What are the component parts? What would it look like? So we would need, and okay, my pointer is red, the background is red, that is an interesting one. And you would need some form of a machine that can actually do the work for you automatically, okay? So the more hands-off you are, the better it is. So what you really want is the user to specify some input, give some sort of a specification. I want my material to be able to do this, okay? What is this in the case of the vaccines we're having right now? To be able to instruct the body to make one of the proteins that we find in the virus, not absolutely everything the virus comprises of, but one of the proteins, in fact, one particular protein which is in the spikes of the particular virus, such that the body can be train itself to recognize that this is an invader and prevent it from binding to the cells and causing damage. So that's the specification, right? So there is a way these days to kind of specify this protein. Then, of course, we have to have some sort of a way to put it inside the system and make our specification go through this cycle. Okay, work iteratively, create new experiments, infer what the next experiment is going to be, and create a specification, an actionable specification for the machine. And as we go along, right, be able to assess, evaluate, monitor, measure what it is we're making. That is a big challenge in itself. And at the same time, be able to capture that knowledge, because the thing is, there is a lot of data that can be generated by arrangements like this. So what do we do with it, right? I mean, how can we use it such that we don't have approaches that do some sort of a very basic search, and every time they don't use prior knowledge, you know, going it blindly, but rather use the prior knowledge to make things more efficiently and speed things up. All right, okay, one might think, why do it? Okay, why do all that? I mean, it's all there, isn't it? Htrz, you did mention we do have these high throughput robots. We do have computers. We can work with computers independently. Why we have to go through this trouble and have this super, I mean, automated system? Right. There are two key scientific motivations. Okay. In fact, there is one more scientific motivation. The other one is more of an engineering motivation. So the first one is to confirm our understanding. So if we have a machine that is able to reproduce a phenomenon, that means we ourselves can understand that phenomenon. If we cannot do that, then do we really understand that phenomenon? In other words, you know, making these vaccines or making a particular class of chemicals. Right now, how it's done, there is a lot of serendipity still involved in discovery, right? And no doubt everybody has heard of the fact that a lot of discoveries have been done accidentally, right? This is true. It is still true to this day. But the thing is, if we could automate, then our understanding would increase. And the other one, of course, is productivity, right? Why would we have, to this day, highly experienced people, highly paid scientists, spending their entire day pipetting liquids from one little vial to another? Is this the best use of our time? Of course it's not. Okay, but then the question is, if we were to engineer such a machine, let's start breaking down into its component parts. We would need, one of the things we would need is to make things faster, rapid execution. Why? Because we want to respond to markets fast. And with the vaccines, I mean, we have been really good at it. I mean, we have demonstrated that, especially for certain types of or vaccine technologies, we have been really good at it. Okay, we have managed to, within a year, to come, or less actually, from the time somebody sequenced the genome of the virus to actually having the first vaccine. It was a breakneck speed. Okay, so that's really important. Then it is execution as well. As we mentioned before, we really want the productivity, but we really want to make it at a low cost. And of course, can we control it to make sure that we meet the specs at all times? No batch crashes. Can we do it day in, day out, continuously? No batch crashes, meeting the spec all the time. Right, so these are the requirements. And in terms of having an automated system, we want a rapid, reliable, repeatable techniques for making. The execution needs to scale. And automation, of course, is highly desirable. We don't need, people pipetting or transferring liquids around. And even to large scale in large biotech plants, this is exactly what is happening to this day. Even all the vaccines we hear in the news today, there are many manual interventions still. And of course, direct control, monitoring, and feedback. And then the question is, okay, if we have all that, can we start thinking in terms of artificial creativity? Can we actually have at some point, the machines actually making crucial decisions without humans in the loop? And this is actually one area of research in artificial intelligence that is finding its way in the space we're discussing right now. All right, okay, so how might a robot scientist look like? One can say, could look like this. Of course, I'm not so sure if this is going to happen in my working lifetime. I don't know. It would be awesome to have really thinking machines. And of course, the actual kind of holy grail is to have somebody's knowledge and being downloaded onto a computer and become much more efficient and be able to handle all these machines without the human biology and the human body getting in the way, right? And I'm sure a lot of you have seen the particular film I am referring to. All right, so we know it is not going to look like this anytime soon, but what is it we can do right now? Okay, we can, of course, turn our attention to developments in artificial intelligence. We can have automated optimization and get inspirational use, actually, of advanced algorithms like reinforcement learning, as I'm showing here right now. So have algorithmic approaches that interact with the environment and have specific functions embedded such that the algorithm itself can evaluate what the physical environment has actually made and be able to make a decision on the next action. This is very possible these days, and I'm sure a lot of our people in the audience today will have come across or even worked in these areas. But the thing is, the novelty, can we apply it in that space? Okay, the question is, right, okay, we could, but question is, what would the actual platform look like? Can we use existing approaches? The answer is, of course, nobody stops somebody to use existing approaches, that the batch-based approaches that everyone is using in practical industrial applications today. But the thing is, there is more to life than these existing approaches. So let's look at new and exciting techniques in the space of process intensification, which is really defined as integrating as many of the unit operations, as they're called, as you possibly physically can, unless there is a physical constraint, and try to make them more efficient by, for example, in our case, making sure that a heat and mass transfer, which are very, very important, are done very efficiently. And the challenge is with batch, of course, there is heat and mass transfer there as well, because otherwise we will not be able to make anything. But it is so complex, it is so hard to do, and so prone to risk, right? That it is expensive, and we don't always get the consistency that we want. And also, we have to do a lot of work downstream after the actual synthesis that has taken place to clean up all the mess from all the byproducts that have been created. So the thing is, can we use different geometries, smaller volumes, but have these pieces of kit, this piece of equipment work efficiently in terms of heat and mass transfer, but at the same time work continuously? So if you had a continuous stream, you really don't need these big buckets, which you have to fill them up and then decant them and then start all over again. You want to set up a device and having it run at all times without interruption, unless you press the button stop. So this is the kind of work on the hardware side we have been very, very heavily involved with. And there are these machines actually coming on stream, coming to production in various areas in the space of chemical synthesis in particular, and in pharmaceuticals. There are university spin-out companies that are involved in that, as well as a little bit more established companies who offer such kit. But the thing is, if somebody were to combine these with the kind of intelligent automation and the algorithmic approaches we discussed before, then we could have a situation where you have the machine, you have the algorithm, you do the discovery, still at a small scale, but then you can switch the button and you can start making continuously. Once you have your product, you also have your process. So that means a massive reduction in the time to market with these approaches. All right, so let's have a look at some examples to date. In terms of algorithms, I did show actually very advanced algorithms by means of the reinforcement learning, very, very exciting stuff, but not all materials, not all situations lend themselves to that level of complexity, nor it makes economic justification. So we can use other types of math or systematic approaches that are underpinned by math, such as the design of experiments methodologies, first developed by Taguchi when they were designing the Toyota manufacturing system back in the 60s, I think, 70s maybe, can't remember. And then their evolution into full optimization techniques by means of response service methodologies, whereby we strategically select the points we want to test by means of an experiment. We don't test absolutely everything that there is out there. And then using math to figure out how to create a potential solution space mathematically, and then navigate through that space. So using these techniques in mind, we have worked together with the University of Bath, the EPSRC, the Research Council funded Direct Assembly Network, and another very significant investment by the UK Research Council on metastable materials to actually scale up by these means one very innovative material, not very much in the space of therapeutics, but I just want to give you an idea of what is possible. This material has very interesting properties. It changes some fundamental physical properties by means of its size, and also by the application of some external stimuli, heat, for example. So the intention of these materials is to use them as temperature sensors without being a thermometer and having all the complexity. So have smart coating applications. So dialing the size of that particle at the nanoscale is very important, and it is driven by the process. So I have two examples here. One is done by the more sort of traditional batch approaches, and you can see you literally have kind of like a dog's breakfast, as it were, or you could use the much more controlled and yet continuous approach using flow synthesis, where you can clearly see how much control you can have on the particle size and the particle morphology. So you can see a much more consistent product. Right, okay, so to get this to work, we used the flow system that we have developed with a special geometry to enable this enhanced heat and mass transfer, and also we combined it with a mathematical model and to allow us to select properly the kind of size, shape, and morphology out of all the different options, with the view to formulation, okay, how it is going to be formulated into a coating. Trouble is, if you use these approaches, then you can mathematically get trapped in just a small sort of subset of the possibilities that you might have. Because any mathematical function that is complex, that's not a very complex, but imagine a multidimensional space, you can have these peaks, you can have these points here, these troughs, but then there is only one global maximum and one global minimum, depending what it is you're searching for. And if you use methodologies like the one I have shown, you could get trapped into a situation like this, into an area like this, into an area like this or this, depending whether you do maximization or minimization, and then you are missing out on a solution that is really, really good. So how can we improve on that? Again, what we did was we used artificial intelligence, evolutionary optimization strategies in particular, genetic programming, and I'm sure a lot of people in the audience will have come across these. And so the idea here is to actually mix and match a bunch of promising solutions and gradually converge to the solution that is optimal. The thing is, by doing this, actually, what you essentially do, you can operate in unstructured mathematical spaces with a very large or even undefined number of parameters. You may not know the parameters a priori, and you also discover the parameters as you go along. They're very easy to develop, they're very robust, and importantly, they're very suitable when you have hardware in the loop. Okay. The other crucial thing is that they allow global search, which avoid getting trapped in local regions. All right. So, I mean, this is a pictorial representation of what I mean. You could have a situation that is, you know, you have a space that looks like this. And whilst you could get trapped in the green area here, if you start pulling yourself out using these methodologies, you can then find yourself or drop yourself into a better region that converges faster to the optimal. And the thing is, you would then need to figure out how to integrate with the hardware. And integration here means really finding out what it is to measure and what can you measure online to have the full closed loop directed automation that I have been alluding to. Right, let's see very quickly an example of that. So this is a small drug-like molecule. Okay, the interesting thing about this molecule is that it has one intermediate here that is a fluorophore. That means if it gets stimulated by some external light source, so if we have photons incident on it, then it fluoresces, it emits photons. The actual final product does not, right? So that's a very good way to do two things. Number one, to allow us to test the methodology by switching from the fluorophore to the final product, selecting which one to make and when to stop the reaction, and of course, making sure if we are interested in the final product, to make sure that we stop at the right moment. Right, okay, so based on our experiments, if I can make the slides change, we did use a flow system, two types of flow system and a batch system. And what you can see here, although in the flow system, you have a very nice peak of the fluorophore, we were interested in stopping the reaction at the fluorophore. And the system at the bottom with a more efficient mixing compared to the one at the top, and therefore a more efficient process from the manufacturing perspective, was converging much rapidly or finishing much rapidly, which meant that the reaction was much faster. We're able to intensify the reaction even further. So we thought, okay, why don't we try to then optimize and maximize the production of the fluorophore inside the faster, better system. So we use these genetic algorithms to start from that promising point and try to find the best solution that we were able to identify in our experiments. And after several iterations, completely hands-off, completely in an automated way, we were able to improve at least 30% on our original point. Now, the interesting thing about that, of course, is from the production perspective, 30% improvement is quite significant. I mean, we would be lucky if we can get 5–10% improvement at a time. So all that was going so well until SARS-CoV-2 happened. And we thought, all right, okay. At that time, of course, or before that, we started working in the area of vaccines with our colleagues, Imperial, Nottingham University, Bristol, and several pharmaceutical companies. But then SARS-CoV-2 happened and we had to retool our work very, very quickly. Thing is, when we started working in that space, we thought, all right, okay, the current process we were looking at, and the current vaccine platform in particular, which was an RNA-based vaccine, and we have heard a lot about RNA vaccines these days. One of those that is being rolled out is an RNA-based type vaccine, and it is made by the techniques that I'm showing right now. So when we started studying this reaction, and I first came across it, I thought, okay, now this looks more like a chemistry problem. So why can't we make it look like one of the problems we have seen so far, okay? Like the ones I have just demonstrated, and use flow technologies and these flow platforms to actually try and make them better, faster, cheaper, and importantly, continuously. But then this also gave us another opportunity, because shrinking down these big vats they currently use to make these materials, with all the uncertainties, risks, and so on, with flow techniques, we can really scale them down, really a lot, right? And at the end of the day, we only need just a couple of milligram per milliliter or liter even, in terms of the quantity of the therapeutic itself. So the concentrations are really low. So I thought, okay, right, why don't we then think in terms of a factor in the box? Can we make these materials at the point of use? And if we were to do that, what would we need? So after a lot of design iterations, going back and forth and thinking, we came up with the idea of why don't we use techniques that are used extensively, very scalable in the semiconductor manufacturing business for the last 50 years or so, right? Let's use these techniques and create a chip type reactor system, flow reactor system, that we can control very, very precisely the conditions, the heat and mass transfer, that is, by making the geometry suitable, and of course making sure that there is a consistent heat going in, and essentially mimic what happens inside the cell to make these materials, because our bodies make these RNAs all the time using exactly the same technique, but do it at a scale that we can make them at a local hospital and administer these materials at the point of use. So even going beyond the vaccines, one can start thinking about nucleic acid therapies to treat truly personalized diseases, such as genetic diseases, or even cancers and stuff like that, really at the point of use. Right, okay, measurement, I did mention to automate anything, you really need a very good measurement system. We have included in our design, we have included a spectroscopy, which hooked it up to the system online to be able to assess very quickly what we have, and have some sort of a means of at least a qualitative quality control. So you make this material continuously, and then you know with confidence what it is you're making. In our tests, and this is one of our very older setups, actually, in our tests we have been able to compare the final product that we knew of with what was coming out of the reactor, and you could see how we managed to not only keep the same profile, which from a spectroscopy perspective, this is really important, but also, when you see peaks going up, that means we're making more of that material. Then, of course, that gave rise to other very interesting questions. What would a supply chain for something like that look like? So if we were to move away from these big batch plants, how would the supply chain look like? Of course, it has to be compliant with good manufacturing practices, regulation, and so on. But then what we would have to do actually is really distribute the device itself, which kind of looks like a big chip with everything in it, so the material can be made on site. Now, the question is, does this make commercial sense? And if I can make it work, oops, sorry about that. My system is a little bit slow. can we make it commercially work? How would such a supply chain look like in comparison to the big existing batch plants that we see here? Would the economics work? We did indeed develop mathematical models, mathematical optimization models, exploring the entire supply chain. We have some early data. We do carry on with that work, but we have figured out that based on our analysis, right? If we were to have such an arrangement to vaccinate the entire population of England, we would need 60 sites across the UK, and we would be able to vaccinate the entire population of England across these 60 sites within a couple of weeks, given the productivities and the localized manufacturing arrangement that we have. In terms of costs, because one saves a lot on the very complex current supply chain, which is a cold chain, very expensive, and also on labor and inventory, and the fact that you can make it on demand, one can see the costs per dose dropping to less than $1. We did the calculation in dollars because this industry primarily talks with dollars, unfortunately, or fortunately. Right. Okay. I know I'm quite a bit over the time. Just in summary, I have shown really a cross section of the stuff we have been doing. You can see at my back the actual kit we use to make these reactor systems and to test at the moment. These are techniques that are very widely cheap to run and very well established with a very high level of repeatability. I have shown you how we can make various chemical systems as well as RNA-based vaccines with this new manufacturing sort of automation type thinking. And, looking at how we might take these technologies forward to actually deploy them quickly and serve large populations much faster and at lower risk than what we're currently doing. Of course, there is always a danger by various stakeholders, by various bodies that they might actually consider this like the situation we see here in the cartoon, which is, I mean, it's a very nice cartoon. I did nick it off the web, as you can imagine. But it kind of really describes some of the attitudes towards new technologies, especially in that space. But we're working towards going around this problem. Okay, acknowledgements, very big team. My previous, current postdocs, PhDs, researchers, and also colleagues from the various institutions we have been working with in the work that I have demonstrated. The list is quite a lot longer than that. Right, so at this point, I would like to thank you very much for your attention and your patience. I know my apologies for going over time, but there was quite a lot of exciting stuff I wanted to share with you, at least things that I found exciting. I very much hope that you can share the sentiment. So thanks very much for your attention. And I think now we are ready for questions. So I don't know, Agnes, shall I stop sharing my screen, or David, do I stop sharing?
Yes, I think that's right. That's okay. That's it. I think people can see me, and maybe that's good or bad. But of course, you can see me, and I'm very much lost for words because you've covered so many different aspects of process engineering, chemical engineering, mechanical engineering. And I think the fascinating thing that you've prompted in my head is that you come up with the sort of transfer technology, if you like, of miniaturization out of the semiconductor industry, which is quite fascinating. And so we'll probably just kick off with a question or two. I can see that there are one or two that have come in. So on a personal level, I've worked with a lot of process engineers and I in the petrochemical industry,
right?
And I can see that they were, justifiably much faster brains than mine. But within your chemical engineering background, did you have a sort of light bulb moment when you said, actually, I want to focus on research of a vaccine manufacturing process? Was there a sort of moment that it clicked and you said, yes, that's what I want to do?
Yes, actually, there was. Indeed, there was. And in fact, I distinctly remember the moment, right? It was just at the initial phases of the vaccine project we started about 4 years ago now. And we were in one of my collaborators' office, and we were discussing about their approach they were taking on the RNA vaccines and so on. Although in the project we did have a number of vaccine technologies, not just RNA, but a few others as well. And it sort of clicked. And when he carried on speaking, I thought, hang on a minute. I mean, that is a chemistry problem. It's not a synthetic biology problem, right? So can we then use what we knew already about the work we have been doing in the past, like the one I have shown, and make it look like a chemistry problem, and perhaps scale it up quickly? Because they told me we're working, on these microliters, how we can scale up, and I mean, we know we can do all these nice things. And I thought, okay, leave it with me, right? And on the way back in the tube ride, I did come up with the idea to use these chips and specific geometries. We started working out, doing several iterations, and there we are. SARS-CoV-2 happened, and it was all very relevant all of a sudden.
Thank you very much. Yes, obviously it did certainly inspire you. Very good. I'm going to hand over to Bobby.
Hi, thank you very much for that really fascinating talk, Htrz. We got one question from one of the audience, and I thought I would ask that in conjunction with another question that I had, because I think you could maybe answer them together, or certainly they're very related. Yeah. So one of the questions we've been asked is, what is the current position of using this technology for COVID vaccines? And secondly, perhaps rolling on from that, it's very relevant, is how easy is it for the factory in a box system for vaccine manufacture to be deployed in developing countries, or remote or rural areas, which might have poor accessibility to, let's say, electricity or power. And then I think you touched on it at the very end, but perhaps you could elaborate a little bit on what are the next steps for developing the factory in a box.
Yeah. Okay. Really, really good questions. And the entire project we're engaged with on vaccines was initially conceived for coming up with new manufacturing methods that are deployable in low and middle income countries. So that was a starting position. Then we did, and I see on the chart, RNA was one option, and we're looking at various diseases that you see in certain geographies, such as rabies, Ebola, chikungunya, and other things. Some of them are more like, including HIV, some of them are more like textbook type test grounds. Some others are more like very, very important to solve. RNA was one of the candidates, and then SARS-CoV-2 happened. So the entire team, not just us, the entire team retooled our research on that subject only, or on this pathogen rather, only. And our synthetic biology and medical colleagues did the sequencing and created the one of the components that go into the process, the plasmids, and then we kept working on the stuff we were working already anyway. So we in particular were very well placed to carry on with the research. So in terms of where we go from here, I think the sky is really the limit, because what this pandemic has demonstrated, at least for the RNA space, is that it is going to be a very, very viable form of therapeutic, really cutting-edge therapeutic that is not just me, but I strongly believe it is going to revolutionize medicine. So having the manufacturing techniques that you can make rapidly, on demand, and reconfigure manufacturing at very, very low risk one can address a very wide range of diseases. So where we're heading from here is to focus almost entirely on this research, on this work, continue with the engineering, and the important crucial step for us right now is regulatory compliance. How we can engineer regulatory compliance out of the box. So that's what the focus is. The good news is our MHRA here in the UK, they are warming up to the idea of distributed manufacturing. There are still challenges, but they are very warming up to this idea. So this is really good news for us.
Now we have a question which has come in. And I think it's a good one. Is you did talk about measurement and all the sort of control systems you need, but how can you be sure that you've got the same quality throughout all those dispersed locations? I mean, you talked about 60 sites in England. How can you really, is there a centralized way of making sure the quality is there?
Yeah, right. Okay. So this is really the engineering I was talking about. So we can do nice qualitative measurements right now. There is a lot more work to be done in that space. There is ways, without wanting to give away too much, because otherwise, where's the fun in that? I mean, come back next year. So there are ways of ensuring quality having an open loop approach, because you cannot really measure everything in line. You would have to have some measurements that are done offline. So if you make sure that you have this consistency and repeatability at the process level, and that does not change, then you can have a high level of quality assurance, provided that you also have additional measurements without having to go to the whole shebang of all the measurements and analytics you would have to do. This is the thinking, not just our thinking, but it is the thinking of industry as well in other and adjacent domains. So there's a lot of work that's going on in that space.
I'm just following on. I think we actually have a question from Professor Dola. And he's asking, he wants to know about how your methodology can be used in the context of finding new biodegradable or compostable materials. Perhaps you can elaborate.
Yeah, right. Okay, this is okay. My title is Professor of Sustainable Manufacturing, and I should really actually be working in that space. Misha is absolutely right for reminding me that maybe I'm going a little bit off the rails. So yeah, I mean, it is quite possible to enable material discovery, and in particular process discovery, which is our space, and the process scale up. That is really our space using these approaches. We have worked extensively with colleagues and also various university spin-outs in the area of growing cells for making a number of things coming out of these cells, from biomaterials to actually fuels and stuff. So we have gone through this process. We have shown it is possible. And we have one PhD published in that space, and one MSc thesis published in that space. A lot of that stuff is also in the sort of commercial domain. So yeah, really exciting work. But right now is all about vaccines.
Thank you.
Htrz, I'm sort of aware that time is getting a bit close to the amount, but we've got questions.
You've muted yourself, David.
Yeah, I think it's a case of saying there are probably loads of more questions that may come up, and we may have to sort of allow those to be followed up later. But I do want to just ask one quick question, and that is if you've got some advice to people maybe listening in who are thinking about moving into research or being entrepreneurial indeed, because your experience has got a lot of industry transfer I can see in it. Is there any advice that you can sort of give those people thinking, and also how did King's help you in that sort of development of research and transfer technology?
Right, really, really good question, and this is a subject that is very, very close to my heart. As you said, David, I have been involved in technology transfer for a number of years, actually, nearly 20 years now. I had gone through the spin-out process. you know, entrepreneurship, especially technology entrepreneurship, is all about solving big, difficult problems. That's what it is all about. It is about money to some extent, but I still think it is more about solving these problems that touch upon the lives of many, many individuals, be it, you know, consumers or you know, businesses or whatever. So it kind of sounds like being also an academic researcher, because in academia we do exactly the same thing, right? With less funding perhaps, but you know, anyone, I would not change this career for anything. It does give a lot of opportunity, a lot of options, a lot of freedom to think, invent, innovate, and it does enable you to bring your, if you want, your ideas all the way to market if you can. The process is difficult. The UK universities, however, are very well positioned and geared towards technology transfer. So we have offices and specialists at King's and other universities, but King's, of course, being, you know, one of the top, who actually can help somebody with the process. Students, there is specific student support for that, but also, of course, academic staff who can, if they're interested in, follow this route.
Well, thank you. And I think it's fair to say we are sort of at the end of time allocated, which I know you've stimulated lots of thoughts. I can see more and more questions. But thank you very much, Professor Harris, for giving us that, you know, that opening, that eye-opening, that little peek, I should say, into the opportunities for scalable pharmaceutical manufacturing, and it's fascinating. I just also would like to thank Agnes and the team at the Alumni Office for everything they've been doing in the background and getting this whole webinar set up for us. And I do want to thank Bobby there as our event secretary for getting the word out so efficiently in a way that I couldn't possibly do. But as a quick heads up for those alumni who are familiar, we normally have an annual lunch held on campus in early June. Well, again, this year, we're not going to be able to do that. However, I just want to let you know that we are searching for a suitable date, which we hope will happen in the autumn term, you know, subject to all the things that are obvious with the virus being under control. I think finally, I want to thank all of those of you who've joined us this evening, who've participated in this evening as well. And if you'd like to learn more about King's work on this topic and updates on the engineering department itself, please make sure to let us know. After this event, you'll get a survey, so then resources can be shared with you. So I think that's it. I will close now my words, but again, many, many thanks to Professor Harris for all that you've stimulated in our minds. Thank you very much.
Thanks very much, everybody. It was a pleasure and a privilege.
Thank you for everyone joining.